Hi everyone. In this lecture, we will be talking about our different labeled immunoassays. So just a short overview on what labeled immunoassays are. They are designed to detect antigens and antibodies that may be small in size or present in a very low concentration. They use labeled reactants in order to indirectly detect the target. And instead of detecting the actual antigen-antibody complex, the presence of the target is indicated by detecting the specific binding of the labeled reactants instead. In a labeled immunoassay, the analyte is referred to as the substance to be measured, and it can be an antibody or an antigen. There are 4 types of labeled immunoassays. They have very similar principles, and they only differ in the label that is being used in the assay. We have radioimmunoassays, enzyme immunoassays, fluorescent immunoassays, and chemiluminescent immunoassays. We can classify our different labeled immunoassays in many ways. The first classification is based on the format of the labeled assay. We have 2 classifications. First, the competitive immunoassay and the non-competitive immunoassay. In a competitive immunoassay, patient and reagent antigens compete for limited binding sites. The keyword here is that there are only limited binding sites, and that patient and reagent antigens have to compete with each other in order to bind to those sites. Next, we have our non-competitive immunoassay. Here, the patient antigen is not competing with a reagent antigen, rather it is the only antigen that is directly captured by an abundant number of binding sites. So again, here the keyword is there is an abundant number of binding sites. Let's talk about the competitive immunoassay in more detail. Here are the various reactants used in this assay. We have the reagent antigen, which is labeled, the patient antigen, which is unlabeled, and the reagent antibody. In a competitive immunoassay, all reactants are combined at the same time in the same reaction vessel. The competitive immunoassay is based on this principle. Unlabeled patient antigens compete with labeled reagent antigens for limited antibody binding sites. If a patient antigen is present, some of the binding sites will be filled with unlabeled analyte, thus decreasing the amount of bound labeled reagent antigen. The detected amount of labeled antigen is inversely proportional, therefore, to the concentration of the patient antigen. In this figure, you can see a demonstration of what a competitive immunoassay is. We have 2 setups. On the left are setups without the patient antigen, and on the right are setups with the patient antigen. So let's look at the steps one by one. So all the antigens are added to the test tube or the reaction vessel. If the patient is not positive for whatever target we are looking for, only the labeled antigens are able to bind to the limited binding sites of the reaction vessel. In contrast, if the patient has the target, then some of the target antigens which are unlabeled will be able to bind to the antibodies instead, and fewer labeled antigens are able to bind. So we wash off the excess unbound reactants and measure the signal. If your patient does not have the target, then they will have a very strong signal because all the antigen binding sites are filled with our labeled antigens. In contrast, if your patient has the target, then the signal will be weaker because some of the antigen binding sites are filled up with unlabeled antigens. Here you can see the relationship between the signal and our analyte concentration. They have an inversely proportional relationship, which means that the higher the signal, the lower the analyte concentration. And at the opposite end, the lower the signal, the higher the analyte concentration. Because of the competitive nature of our competitive immunoassay, the detection limits are determined by the affinity of the antibody towards the patient's antigens. So the higher the affinity of the antibodies used in the test for the patient's antigens, the higher the sensitivity will be. Next, let's discuss our non-competitive immunoassay. The reactants here are, first, the capture antibody, which is unlabeled. This is the reagent antibody, and this is placed on the solid phase. Next, we have the patient sample or the antigen and the secondary antibody. Here, this is the antibody that is labeled, or the antibody that gives us the signal in order to detect our immune complex. So the patient sample is added to a solid phase with a capture antibody and allowed to react. After, the secondary antibody is added. Here, you can see the principle of your non-competitive immunoassay. First, primary antibody, also known as the capture antibody, is added to a solid phase. Sometimes, some non-competitive immunoassays use a capture antigen instead of a capture antibody. Next, unknown patient antigen is then allowed to react and be captured by the solid phase antibody. Then, a second antibody with a label is added and attached to the capture antigen. Then, the amount of label is measured and is directly proportional to the amount of patient antigen in the test. In this figure, you can see how a non-competitive immunoassay works. So we have again two samples. The first sample, sample A, does not have the target analyte. Meanwhile, sample B does have the target analyte. So in a non-competitive immunoassay, there is an abundance of antigen binding sites on the solid phase. And this is given to us by the primary antibody or the capture antibody. So in sample A, since there are no analytes in the solution or in the patient sample, then there will be no binding that occurs. And in sample B, since this has the patient analyte, you will be able to find specific binding. After this binding has occurred, we remove or wash off the unbound reactants, and we add our secondary antibody. This secondary antibody has a label attached to it. And this secondary antibody is specific to whatever antigen we are trying to detect in the test. So the secondary antibody binds to these antigens. Here you can see that here. And of course, if there are no antigens to begin with, then the secondary antibody cannot bind to anything. So it is just washed off when we wash our setups. Afterward, we detect the signal of our different reaction vessels. So the higher the signal given to us by the secondary antibody, this means that there is also a very high analyte concentration. So the relationship between the two is directly proportional. Again, the higher the signal, the higher the analyte concentration. As mentioned earlier, the non-competitive immunoassay can also use antigens in the solid phase. So instead of capture antibodies, here you have capture antigens. Now, the antigens bind to antibodies found in the patient's serum, and the process repeats itself. This time, we have a secondary antibody against the patient antibody that we are trying to detect. And if the patient does have the antibodies of interest, then the secondary antibody will be able to bind to them and produce a signal which we can detect. And again, the relationship is still directly proportional. The higher the signal given to us by the secondary antibody, the higher the analyte concentration. In this case, this is the antibody concentration in the patient sample. The next classification for our labeled immunoassays involves the need for separation steps. There are two classifications. First is the homogeneous immunoassay, and next is the heterogeneous immunoassay. In a homogeneous immunoassay, the test does not need a separation step. This means that a homogeneous immunoassay is usually a one-step test. This usually only uses a liquid phase, and although it is more simple to perform than the heterogeneous immunoassay, it is less sensitive. Next, we have the heterogeneous immunoassay. These kinds of tests include a step to physically separate free from bound analytes. These are what are known as washing steps. Heterogeneous immunoassays usually employ capture antigens or antibodies that are attached to solid phase by physical adsorption. Let's now talk about each of the different labeled immunoassays in more detail. First, we have the radioimmunoassay, or RIA. This was the first type of immunoassay to be developed, and it was developed by Yalow and Burson in the late 1950s. This kind of test is very useful for detecting substances that have a very small concentration in a sample. They use radioactive substances as their label. That's why it is called a radioimmunoassay. The most common type of radioactive substance used is iodine-125. Now, our radioimmunoassays are very precise and sensitive, but they have inherent disadvantages. First, radioactive labels are considered a health hazard, and they also have a relatively short shelf life. For example, our iodine-125 has a half-life of 60 days, so you have to use the reagents in a very short amount of time. Radioimmunoassays also use very expensive equipment, and the equipment for radioimmunoassays can really only be used for this type of test. So it is not very practical if you wanted to perform a wide variety of tests. For these different reasons, our radioimmunoassay has been replaced by chemiluminescent reactions in most modern laboratories. Here you can see how the radioimmunoassay works. First, you have your labeled and unlabeled antigens. Here, the labeled antigens are labeled with the radioactive substances. And of course, the unlabeled antigens are our patient antigens. We add this to a vessel with our solid phase antibody. So we have our specific binding phase. Our different antigens compete with each other in order to bind to limited antigen binding sites here on our solid phase. So this is an example of a competitive immunoassay. Then we have a wash step. Because of this wash step, the radioimmunoassay is also what is known as a heterogeneous immunoassay. So the wash step removes any unbound reactants. After, we then analyze the signal given off by our radioactive labels. So the higher the radioactivity, the lower the target concentration. Because again, if there is a low concentration of patient target, our labeled antigens have more room to bind to our different antigen binding sites. In contrast, if you have an abundance of unlabeled antigens or patient antigens, then when you add them to your solid phase antibody, they have to compete with our labeled antigens. Usually, the test should have a higher affinity for the patient or the unlabeled antigens. So in this figure, you can see that only a few radioactive antigens are attached to our different antibodies. When we wash off the unlabeled reactants, only a few remain attached on the antibody. So when we detect the signal, there will be a lower radioactivity signal. And this means that the lower the radioactivity, the higher the target concentration. Nowadays, the main examples of radioimmunoassays being widely used are your radioimmunosorbent assay or RIST. This is a test that is used to measure total IgE, and our radioallergosorbent assay or RAST. This is used to measure IgE towards specific allergens. If you recall our discussion on antigens and antibodies, IgE is the least abundant type of antibody, and so the radioimmunoassay is very useful in detecting them. The next type of immunoassay we will be discussing is called the enzyme immunoassay. As the name suggests, this type of test uses enzymes as labels instead of radioactive material. Enzymes are molecules that catalyze biochemical reactions, and they react with suitable substrates to produce either chromogenic, fluorogenic, or luminescent breakdown products. When we say chromogenic, this means that the compounds give the solution a specific color. Fluorogenic compounds are compounds that, when excited by light, give off light in turn at a lower wavelength. Meanwhile, luminescent compounds are compounds that are able to give light by themselves without the need for excitation. Some examples of common enzymes used today are horseradish peroxidase, otherwise known as HRP, alkaline phosphatase or ALP, and beta-D-galactosidase. Among these three, HRP and ALP are the most widely used. What are some of the advantages for our enzyme immunoassays? First, they can be used for qualitative and quantitative measurements. When we say qualitative measurements, this means that the test just tells us if the patient has or doesn't have the target of interest. Meanwhile, quantitative measurements will tell us the exact concentration of the analyte of interest. Also, the next advantage is that enzymes are cheap and readily available, and they have a relatively long shelf life in comparison to our radioactive materials. Enzyme immunoassays are also very adaptable to automation, and they can be measured with inexpensive equipment. Some enzyme immunoassays can even be measured with just the naked eye. Enzyme immunoassays are also highly sensitive. Next, let's talk about the different criteria that are used to select the enzymes for our enzyme immunoassay. First is the number of substrate molecules converted per molecule of enzyme. This tells us how fast the enzyme can work and how much signal each enzyme can provide us. The next is the ease and the speed of detection. When we say ease, this tells us how easy it is to perform the test. How many steps does it need? Does it need a variety of holding temperatures? And next is the speed of detection. How fast can we get a positive or a negative result? Next, we have the stability of the enzyme. This tells us if the enzyme needs specific storage requirements, like if it needs to be refrigerated, or does it need to be frozen, or can we store it at room temperature. Next is the availability and cost of the enzyme and the substrate. Both of them need to be cost effective in order for the test to be feasible. Let's now move on to the different types of enzyme immunoassays. The first classification are the heterogeneous enzyme immunoassays, and under that we have the competitive enzyme immunoassay. This was the first enzyme immunoassay to be developed, and it is similar to our radioimmunoassay in principle. Typically, they are used for measuring small antigens that are relatively pure, like your drugs and hormones. Let's now take a look at the principle for our competitive enzyme immunoassay. This is similar to the principle of our radioimmunoassay, but instead it uses different labels. So here, we still have labeled and unlabeled antigens, but the labeled antigens are labeled with an enzyme rather than a radioactive material. And of course, our unlabeled antigens are the patient antigens or the antigens of interest. We add this to our solid phase antibody, and competitive binding occurs. So both the labeled and unlabeled antigens compete for limited antigen binding sites here in our reaction vessel. Of course, the antibodies should have a higher affinity toward the unlabeled or the patient antigens. We also have a washing step, which washes out the unlabeled antigens. After, we add the substrate. The substrate then reacts with our enzyme and produces our signal. So we detect this signal, and this tells us how much target is in the solution. The higher the signal we detect, the higher the target concentration. This means that the relationship between the two is directly proportional. Next, we have our non-competitive enzyme immunoassay, again still under your heterogeneous enzyme immunoassays. This is also known as our enzyme-linked immunosorbent assay, or ELISA, and is an indirect method of detecting our targets. This is because instead of detecting the actual immune complex, we actually use an enzyme-linked secondary antibody to bind to the test immune complex, and the secondary antibody is what we actually detect. So this test has a higher sensitivity compared with its competitive counterparts. For the principle of the non-competitive enzyme immunoassay, antigens or antibodies are attached to a solid phase, and here are some examples of solid phases that we are using. First, we have microtiter plates. You can also use nitrocellulose membranes and even magnetic latex or plastic beads. The solid phases, again, are where the capture antigen or the capture antibody is attached to. After, the patient sample is added and the target binds to the solid phase. The enzyme-linked secondary antibody is then added, and this binds to the target or our test complex. The substrate is added and allowed to react with the enzyme. Then we can detect the signal. So that is color, fluorescence, or luminescence that is measured and compared with the amount of product according to a standard curve. Again, the amount of detected signal is directly proportional to the amount of target in the solution. In this figure, you can see the principle of how the non-competitive enzyme immunoassay works. First, we have our patient antibodies, and we add them to our solid phase. Here, the solid phase contains our capture antigen. So we add our antibodies, and they attach to their specific antigen. Now, since this is a non-competitive enzyme immunoassay, there is an abundant amount of capture antigens. So we have a washing step, which removes the unbound reactants, and then we add our secondary antibody. Take note that the secondary antibody here has a label, which is an enzyme. We wash off the unbound reactants again, and then we add our substrate. The substrate reacts with the enzyme and gives off a signal which we can detect. The higher the signal detected, the higher the target concentration. Again, the relationship between the two is directly proportional. Next, let's talk about our capture or sandwich immunoassays. This is just a special term we use for any non-competitive enzyme immunoassay that uses antibodies bound to a solid phase rather than antigen. This kind of test can be used to detect either antigens or antibodies that have multiple determinants. Some commonly detected examples include antibodies. These can be antibodies to specific pathogens, for example. Then we can also detect cytokines, proteins, and tumor markers, and even microorganisms, especially our viruses. Here you can see how the capture or sandwich immunoassay works. So again, instead of antigen attached to our solid phase, here we have our solid phase antibody, and we are detecting our patient antigens. The capture or sandwich immunoassay can again also detect antibodies. So here we add our patient antigens in our reaction vessel, and specific binding takes place. We wash off the excess antigens and add our secondary antibody. Here, the secondary antibody binds to the antigen. This is why it is called a sandwich immunoassay, because the antigen is sandwiched by two antibodies. The secondary antibody has an enzyme label attached to it. So we wash off our unbound reactants, and we add our substrate. Now the substrate interacts with the enzyme and is converted into a signal, either chromogenic, fluorescent, or luminescent. So here you can see that the test vessel now has a signal, and this signal indicates the presence of our target antigen. So the higher the signal, the higher the target concentration. Again, the relationship between the two is directly proportional. Next, let's talk about the homogeneous enzyme immunoassay. Just as a recap, homogeneous assays are one step, and they usually occur in a liquid phase. For our homogeneous enzyme immunoassay, the principle is based on the change in enzyme activity as specific antigen-antibody combination occurs. So here we have an enzyme-labeled antigen and an unlabeled patient antigen. So we have two samples here. Sample A has very few patient antigens. Meanwhile, sample B has many patient antigens. In a homogeneous enzyme immunoassay, the enzyme label used here is actually inactivated when it binds to the antibody. So when it is free, it is active. But when it is bound, it is inactive. So if there are less patient antigens, there is more bound enzyme to the antibody, and therefore more of the enzyme is blocked. And this means that there is less enzymatic activity when we add our substrate. In contrast, if we have more patient antigen, more of them will be able to bind with the antibody, and this means that less of the enzyme is actually blocked because a lot of them are free in the solution, so they can exert their enzymatic activity, and this will give us a higher signal. This means that enzyme activity is directly in proportion to the concentration of patient antigen or hapten present in the solution. So again, the more patient antigen there is, the less blocked enzyme there is, and therefore, the more enzyme activity is detected. For the advantages of our homogeneous enzyme immunoassay, first, it is rapid, and it is also simple to perform because it is only one step, and therefore it does not need expensive equipment. You can also easily adapt it to automation because there are no washing or separation steps. Homogeneous enzyme immunoassays have a similar sensitivity to our radioimmunoassay without creating a health hazard, and their reagents are inexpensive and have a long shelf life. But they can also have some inherent disadvantages. First, they are less sensitive compared to heterogeneous assays. Second, there are a limited number or limited types of enzyme that can be used for our homogeneous enzyme immunoassay. This is because not all enzymes are actually inhibited when they are bound to an antibody. Next, the size of the enzyme label may also be a limiting factor in the design of some assays. Next, some specimens may contain natural inhibitors, and nonspecific protein binding can also occur. These last two are a problem because the homogeneous enzyme immunoassay does not have a washing step, so if there are interfering factors, they will always be present in the test. There are a few factors that affect the sensitivity of our homogeneous enzyme immunoassays. First is the detectability of the enzyme activity. This refers to how strong or how weak the actual enzyme is when in solution. Next is the change in that activity when the antibody binds to the antigen. So ideally, the antigen with the enzyme label should be totally inhibited when it's bound to the antibody. Next is the strength of the antibody's binding towards the antigen, and the susceptibility of the assay to interference from endogenous enzyme activity, cross-reacting antigens, and any enzyme inhibitors that may be present in the patient sample. Because of all the different disadvantages and limitations which we just discussed, the homogeneous enzyme immunoassay is usually only applied to the detection of small molecules that could not be easily measured with other means. Because of course, the homogeneous enzyme immunoassay is far less sensitive than our heterogeneous assays. So the homogeneous assays are usually a last resort test for us. Next, we have our rapid immunoassays. This is still a type of enzyme immunoassay, but it is distinguished by a variety of characteristics. First, it is membrane-based. It usually uses a microporous nylon or a nitrocellulose as its solid phase. Here, the antigen or the antibody can be coupled to the membrane in order to produce the result. They are also easy to perform because they are single-use, disposable assays in a plastic cartridge. Because of this, they can be used for point-of-care testing or home testing. They can also give very reproducible results because they are only read by looking for the presence of a colored reaction product. So it is very easy to read our rapid immunoassay. One of the most common rapid immunoassays being employed today is the immunochromatography kit. This is an easy-to-use one-step kit that allows for the qualitative detection of targets in a patient sample. In this figure, you can see a cross-section of an immunochromatography kit and its different parts. First, we have our sample port and sample pad. This is the area in which we drop the patient sample. The entire kit is composed of a membrane with an absorbent pad at the end. The absorbent pad creates a capillary flow from the sample pad all the way up to the end and is responsible for absorbing excess liquid. Next, we have the conjugate pad, which will contain the labeled antigen or antibody that will form a complex with our target analyte. From the conjugate pad, the immune complexes travel towards the detection zone, where you can find the test line and the control line. These lines contain immobilized antibodies that capture the immune complex. And when the immune complex is captured here, it produces a colored product that can be seen with the naked eye. Going into more detail about how immunochromatography works, first the sample is loaded in the sample port area, and using capillary flow, it moves throughout the membrane. The first thing that happens is that the sample reconstitutes the labeled antigen or antibody, and the two form a complex that migrates toward the detection zone. So this complex is formed here in the conjugate pad, and when the complex is formed, they travel towards the detection zone, which is here. Now, on the detection zone again, there is an antigen or antibody that is immobilized in the different lines found there. And these antigens or antibodies capture the immune complex and forms a colored line for a positive test. which you can see here in this figure. And finally, the excess labeled immunoreactant and liquid migrates to the absorbent pad where they are stored. Next, we will discuss fluorescent immunoassays. As the name suggests, this type of assay uses antibodies that are labeled with fluorophores or fluorochromes. These two terms are interchangeable. Now, our fluorophores or fluorochromes are compounds that can absorb energy from an incident light source and convert that energy into a light of a longer wavelength and a lower energy. This happens as the excited electrons return to their ground state. Each fluorochrome has a characteristic optimum absorption range and should exhibit high intensity and stability in order to be used in a fluorescent immunoassay. One of the most common fluorescent immunoassays is called the immunofluorescent assay. This is the fluorescent detection of antigens on fixed tissue sections or even in live cell suspensions. And in the laboratory, this is used for the rapid identification of microorganisms in cell cultures or infected tissue, tumor-specific antigens on neoplastic tissue, transplantation antigens, and even our CD antigens on T and B cells through the use of cell flow cytometry. Now, among the different fluorophores or fluorochromes, there are two that are mainly used in the laboratory. And these are fluorochromes based on fluorescein and our rhodamine. These fluorochromes are usually conjugated to thiocyanate. And for fluorescein, the most common compound used is fluorescein isothiocyanate, or our FITC. This is a fluorochrome that maximally absorbs energy at 490 to 495 nanometers and emits a green color at 520 nanometers, as you can see in this figure. Next, we have our tetramethyl rhodamine isothiocyanate, or TRITC, and this fluorochrome absorbs light at 550 nanometers and emits a red light at 585 nanometers. Next, let's talk about the different types of immunofluorescent assays. First is the direct immunofluorescent assay. This uses an antibody that is conjugated with a fluorescent tag, and this antibody is directly added to an unknown antigen. This is usually used for rapid antigen detection in tissues or body fluids. In this figure, you can see an example of a direct immunofluorescent assay. Here, the patient antigens are found on tissue that is mounted on the slide. And we directly use fluorescent labeled antibodies to bind to these different antigens. In this assay, the amount of fluorescence detected is directly proportional to the presence of patient antigen on the sample. Next, we have the indirect immunofluorescent assay. In contrast to our direct immunofluorescent assay, the indirect assay uses a fluorescent-labeled secondary antibody. This assay is more sensitive and produces better staining because multiple molecules can bind to each primary molecule of the antibody, thus making this a more sensitive technique. And this can be used for both antigen and antibody detection. In this figure, you can see the principle of our indirect immunofluorescent assay. It is similar to a non-competitive enzyme immunoassay, but it uses a fluorescent-labeled secondary antibody instead of an enzyme-labeled one like you will find in the enzyme immunoassay. So here we have a slide or a solid phase that is adsorbed with the antigen. Then we add the patient sample, which in this case is the patient antibody, and this is captured by the capture antigen. Then we add our secondary antibody. Again, this is conjugated with a fluorescent label. Just like in the direct immunofluorescent assay, in the indirect assay, the amount of fluorescence that we detect is also equivalent or directly proportional to the amount of target in the patient sample. Both of the assays we just discussed, direct and indirect immunofluorescent assays, are examples of heterogeneous immunofluorescent assays. This time, we will discuss the Homogeneous Immunofluorescent Assay, known as Fluorescent Polarization Immunoassay, or FPIA. Just like with all homogeneous assays, this test only has one incubation step and no wash step. And it is also a competitive assay, in which labeled antigens compete with unlabeled antigens in the patient sample for a limited number of antibody binding sites. In a fluorescence polarization immunoassay, the way we detect the concentration of the sample is by detecting changes in the levels of fluorescence polarization. Normally, light travels in a variety of planes, but when it is polarized, it only travels in one plane. And when polarized light hits a fluorochrome, the fluorochrome also produces polarized fluorescence. Now, our labeled antigens can only retain their polarization if they are bound to antibodies. Once they are unbound, they lose their polarization because in a fluorescence polarization immunoassay, the antigens that are usually used are very small, and therefore, when they are in solution, they begin to rotate or tumble. The degree of fluorescence polarization that we detect in an FPIA is inversely proportional to the concentration of an analyte. Because of the nature of FPIA, it is limited to detecting small molecules that are able to tumble freely in solution. Usually, these are our low molecular weight analytes. Aside from that, FPIA also has some inherent disadvantages. Because there is no washing step, there is a chance of interference by autofluorescence produced by different organic substances normally present in the serum. In addition, FPIA also requires expensive and dedicated equipment. This figure shows how fluorescence polarization immunoassays work. Here you can see that if your patient has very little target antibody, a lot of labeled antigens are captured by your antibodies. And when these labeled antigens are excited by polarized light, they also produce fluorescence that is also polarized. And this is detected by a special detector. In contrast, if you have a lot of target analyte in the patient sample, then you will find that more labeled antigens are unbound and free in solution. Because of the small size of these different antigens, they freely tumble in the solution. And when polarized light excites the fluorescent probes found on those antigens, because of the tumbling nature of the antigen, the fluorescence produced is no longer polarized. So once again, in a fluorescence polarization immunoassay, the amount of fluorescence detected is inversely proportional to the concentration of the analyte. The last type of immunoassay we will be discussing today is the Chemiluminescent Immunoassay, or CLIA. As its name suggests, this uses antibodies that are labeled with chemiluminescent compounds. Now, chemiluminescence is the emission of light caused by a chemical reaction producing an excited molecule that decays back to its original state. The most common reaction being used by chemiluminescent molecules is an oxidation reaction, and here we have an example of one such reaction. So we have our chemiluminescent molecule. In the presence of an enzyme catalyst and hydrogen peroxide, it becomes oxidized. And as it becomes oxidized, it produces light, which can be detected by a detector. Some examples of commonly used chemiluminescent compounds in the laboratory are luminol, acridinium esters, ruthenium derivatives, and nitrophenyl oxalates. Our chemiluminescent immunoassays have these inherent advantages. First, they have excellent sensitivity, which is comparable to our enzyme-linked immunoassay and our radioimmunoassay. And as mentioned previously in this lecture, chemiluminescent immunoassays have mostly replaced radioimmunoassays. Next, the reagents are stable and relatively non-toxic, especially compared to our radioimmunoassays. These kinds of tests are also quite inexpensive to perform because very little reagent is used. Next, this type of test has a relatively high speed of detection. This means faster turnaround times in many laboratories. Lastly, detection systems are basically photomultiplier tubes, and this makes machines that are able to detect chemiluminescence simple and relatively inexpensive. For the disadvantages, false results may be obtained if there is a lack of precision in the injection of hydrogen peroxide in the test, or if some biologic materials such as urine or plasma cause quenching or inhibition of the light emission. Alright, so that ends our lecture on labeled immunoassays. If you would like to learn more about them, please check out these references. Thank you for listening, and we hope to see you in the next video.